Hello my dear students, this is Dr. Pooja, your biochemistry mentor. Now in this video, we will be discussing about the classification of the fats. Okay, so this is in continuation with my previous video where I had discussed with the classification of lipids, which commonly we call them as fats. Clinically we call them as TGs. But obviously there are different classification of how we have to classify these fats. So if we talk about the classification, so we can write them as lipids also, we can write them as fats also. So today's topic of discussion is the conjugate lipids or the fats. Okay. Conjugate lipids, or we can call them as fats. And conjugate, along with conjugate lipids, we are going to study about derived and miscellaneous lipids. Now, Bloor was the scientist who actually gave the classification of these lipids. So as per Bloor's system of classification of lipids, we classify them into three main categories. One is your simple lipids, which includes the neutral fat or the waxes. Second is this conjugate lipids, and third is derived lipids, or we can say them miscellaneous lipids also. Now if we talk about the conjugate lipids, it is clear from the word itself, conjugate, which means that they have an extra prosthetic group along with lipid part. Okay, so first of all, conjugate lipids. So they will have obviously a lipid part, and along with this lipid part, they will have a prosthetic group which will be a non-lipid in nature. So this prosthetic group, if it is phosphate, right, so if it is phosphate, it will become a phospholipid. If it is a carbohydrate, it will become a glycolipid. But here lipid will be always there. And if it is a sulfate attached to it, it will be sulfolipid. And the most important class of conjugate lipids is lipoproteins. And lipoproteins is that class which comes actually under proteins also and under lipids also. Why? Because depending upon the proportion of lipid or depending upon the proportion of proteins, it comes under both the classes. Right? And these are the four main classes of conjugate lipids. So in this video, we are going to discuss the chemistry, the importance of all these four classes, because in my previous video also, I told you about the main significance of these lipids is the cell membrane, which is the basis of everything in fact. Cell membrane ka structure, if we see, it has a lipid bilayer. Along with that, it has proteins also. No doubt it has carbohydrates also. But lipids form the major fraction of that. Okay. So first of all, let's start up with phospholipids. Phospholipids. Up now see, we know what are lipids. I have already discussed again in my previous video that lipids are simply the triesters of fatty acid and glycerol. Okay. So if I have to represent a fat, I can simply say that this is glycerol, this is one fatty acid, this is second fatty acid, this is third fatty acid. Okay, and if we have to show, I would say the And the linkage here, which is very important, is linkage is ester. And that is why we always say that lipids or a triglyceride is a triester. They are the triesters of fatty acid and glycerol. So you have to just mug it up by heart. What is a fat? Fat is a triester of fatty acid and glycerol, and in technical language, in I would say clinical language, we call them as triglycerides. Okay, so I can say triglycerides are nothing but they are the esters of fatty acid and glycerol. Now here phospho group is there. So what will happen is fatty acid will automatically be there, but along with fatty acid one phosphate will be attached. This will be an extra group attached to it. So what happens here is that, say this is glycerol, or I will just write the structure like this. Right? Here the fatty acid is there. Here again fatty acid is there, but the third carbon atom will be attached to a phosphate. Right? So this becomes a phosphatidic acid. This entire structure we will call it as phosphatidic acid. Now, this phosphate will be attached to a base. Now, this base can be nitrogen containing, means it has nitrogen in it, or it can be non-nitrogen containing. But whatever is there, a base is attached to it, so the entire thing will be known as phospholipid. This is very important to understand. First of all, see, what is a lipid? Lipid is simply triester of fatty acid and glycerol. Okay. Now, if we are talking about a phospholipid, I told you कि lipid तो रहेगा ही रहेगा. Along with lipid, when a phosphate is attached, it will be known as phosphatidic acid. And this phosphatidic acid, when it is attached to a base, whether it is having nitrogen in it, whether it is not having nitrogen in it, but overall it will be known as a phospholipid. And the most important functions of phospholipids are, they are the major components of the cell membrane. They help in cell signaling, they help in you can say cell transportation, cell recognition. Majority of the functions which are related to the cell membrane, they are performed by these phospholipids because they have a charge on them. And we know whenever a charge is there on the cell membrane, it becomes selectively permeable. Okay. So, and they are also the common lung surfactants. That is what we are going to discuss here. Okay. So, you just have to keep this thing in mind because structures are very, very important. Why structures are important and why I put emphasis on the structure is because recently you people have studied chemistry. Okay, so if you can understand with the help of structures, biochemistry will be like a walk for you. Okay, and it's pretty visible from this structure only that we know what is the structure of glycerol. We know whenever alcohol combines with acid, simple water molecule is lost and a ester is produced. Here 3 esters are produced. Here phosphate is replaced by a fatty acid, and this phosphate is attached to a base which might have nitrogen, which might not have nitrogen with it. So if I talk about the functions, I can simply say that the most important function of phospholipids are, they are the components of cell membrane. And I can simply write it as PL. PL means phospholipids, components of cell membrane. Okay. So, if they are the components, they are used as lung surfactants also. Right. Isme you can just remember phospholipid, L for lipid, L for lung. So, they are used as lung surfactants. used for cell signaling, transportation, recognition, and so on. Okay, and again, if we talk about the structure of this, we know that they are having a hydrophobic and a hydrophilic portion also, that we have already discussed, right? Now, coming over to this, now this phospholipid can change the alcohol also, because fatty acid is going to remain the same, but glycerol can be replaced by some other alcohol, right? So, these phospholipids, this is a very, very important part, that these phospholipids, if the alcohol is glycerol, what I mentioned just now, if the alcohol is glycerol, it is known as glycerophospholipid. So, they will be known as glycerophospholipids, right? And if the alcohol is not glycerol and that alcohol is sphingosine, which is again a very, very important unsaturated alcohol. So, you see the alcohol has changed. So, when the alcohol is sphingosine, it becomes sphingophospholipid. So, this is the basic difference, and these glycerophospholipids can further be classified into two again subclasses. One is nitrogen containing, means they have nitrogen in them, and non-nitrogen containing, means they do not have nitrogen in them. Right. So, if we talk about nitrogen containing, the most common examples are phosphatidyl, because phosphatidic acid is there, phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine, and non-nitrogen will have phosphatidylinositol. So, now see what is the significance. Suppose if I ask you about the structure, nobody is going to ask you write the structure of ethanolamine or write the structure of serine. But yes, phospholipid ka structure can be asked. Why? Because of the mode of action of the enzyme phospholipases. Phospholipases is that class of enzymes which act on phospholipids. Very important. Okay? Because we all know that, again, if we talk about the metabolism, we all know something is synthesized in the body which is degraded also in the body. Okay, so whenever something is synthesized and when it has to be degraded, the enzyme required for degradation is absent, there will be accumulation of that particular thing in the body which will lead to the diseases. Okay, so now this is the brief classification of phospholipids. First of all, what are phospholipids? Phosphate plus lipid. So, if we talk about only phosphate, it will be known as phosphatidic acid. Now, this phosphatidic acid, when it is attached to a base, it becomes a phospholipid. Now, this base can have nitrogen in it, cannot have nitrogen in it. Nitrogen containing, the most important one is phosphatidylcholine, phosphatidylserine and phosphatidylethanolamine. And then non-nitrogen mein the most important example is phosphatidylinositol. There is one more, cardiolipin is also there, which we will be discussing now. Okay. And I told you that here the alcohol is glycerol. So, that is why glycerophospholipid. And if the alcohol is sphingosine, they will become sphingophospholipid. Okay. So, now first of all, we are going to discuss the main points. We are not going into that much detail, but yes, examination point of view say these are important. So, what we are going to study if we talk about phosphatidylcholine. So, phosphatidylcholine Commonly, we call it as lecithin. Right? Lecithin is the other name of phosphatidylcholine. And if we say lecithin mein, if the two fatty acids belongs to palmitic acid, it becomes 𝗵𝗵𝗺𝗺. Okay? That means if two fatty acids belongs to palmitic acid, it will be known as what? DPL. 𝗵𝗵𝗺𝗺. Right? That means if I have to draw the structure of this, I can simply say that CH2O, here palmitic acid is there. Second pe palmitic acid is there. And third pe phosphate is there, and this phosphate which is attached to choline. So choline is the nitrogenous base here. Right. So this becomes your phosphatidic acid, this becomes choline, so overall we call it as lecithin. Okay. So this is palmitic acid. And if we talk about the function, this DPL is the most common lung surfactant, and in fact this is the only lung surfactant, I would say. So, and what is its function? It reduces the surface tension. Reduces the surface tension as it lines the alveolar membrane. Okay. So it is lining the alveolar membrane. So during the process of expiration, to prevent alveoli from collapsing, this DPL is the one which is present as a lining. Okay. So lung surfactant reduces the surface tension as it lines the alveolar membrane. Very important. And deficiency of DPL leads to respiratory distress syndrome. It is again very, very important. So deficiency of DPL will lead to RDS, which is a respiratory distress syndrome. That is why lung maturity test is conducted in the fetus, just to check whether the child, when he is born, his lungs are mature enough. That is lecithin to sphingomyelin ratio. Okay, just to check that. Otherwise, there will be a problem obviously in respiration, respiratory distress syndrome will be caused. So it is just lining the membrane. Okay, so this is phosphatidylcholine, commonly lecithin, example is DPL. Second, if we talk about, second is phosphatidylserine. Now phosphatidylserine, everything would be the same. The only difference will be fatty acid will be there. The only difference will be instead of choline, some serine residue will be there. Okay. So I can say that phosphatidylserine will be fatty acid 1, fatty acid 2 and fatty acid 3. Instead of that, phosphate is there which is attached to serine. And if we talk about the functions, it plays a very, very important role in apoptosis. So basically, phosphatidylserine is a marker for apoptosis. Programmed cell death. Very important. Right? So this is phosphatidylserine. Then third is phosphatidylethanolamine. This phosphatidylethanolamine, again everything will be same except serine, ethanolamine will be present, and it is also known as cephalin. And if we talk about the function of this, it is marking, or you can say present in the CNS. So you can just, it's like just a mnemonic, C for syphilin, C for CNS. So phosphatidylethanol is the one which is lining, which is present in the CNS. Okay. So these are nitrogen containing. Then after this, the most important, again non-nitrogen containing, we can quote the example of phosphatidylinositol. Now if we look at the structure of inositol, it resembles glucose. The only difference here is, glucose has a glycosidic linkage, it does not have a glycosidic linkage. Phosphatidylinositol is like this. Rest everything will be the same. No glycosidic linkage is there, right? And this is attached to phosphate. Rest again everything will be the same. So it is like phosphatidylinositol. And the most important function of this is phosphatidylinositol. Commonly we call it as PI3 also, the most important second messenger. So if it is a second messenger, it is helping in cell signaling. Right? Then after phosphatidylinositol, then comes cardiolipin. Now what is this cardiolipin? This cardiolipin, again it is clear from the word itself, cardio, it was first isolated from the heart muscle. First isolated from the heart muscle. Right? And if we talk about the structure of cardiolipin, it is nothing but it is simply diphosphatidyl-glycerol. Diphosphatidyl-glycerol. And the most important point to remember about cardiolipin is that it is the only phospholipid. Only phospholipid which is antigenic in action. Antigenic in action. Very important. Right. Now, if it is antigenic, what is its function? It can recognize the antibodies against Treponema pallidum. Treponema pallidum, which is responsible for causing syphilis, which is again a sexually transmitted disease. So, recognizes the antibodies raised against Treponema pallidum, causing syphilis. Right? And since it was isolated from the heart muscle and it is lining the inner mitochondrial membrane, lining the inner mitochondrial membrane. And deficiency of this deficiency of cardiolipin leads to Barth's syndrome. Barth's syndrome. Right? So these are what? These are the different type of phospholipids, and most precisely phospholipids, or these are the glycerophospholipids. They will be glycerophospholipids, where the alcohol is glycerol and which is further nitrogen containing and non-nitrogen containing. The example of nitrogen containing: phosphatidylcholine, phosphatidylethanolamine and phosphatidylserine. Non-nitrogen will include phosphatidylinositol, right? If we talk about phosphatidylcholine, I told you lecithin is the common name, and lecithin may be the example is DPL, which stands for 𝗵𝗵𝗺𝗺, which is the most common lung surfactant. Very, very important. What is the function of lung surfactant? It reduces the surface tension as it lines the alveolar membrane. I told you that whenever the alveolus, because we know lungs are responsible for respiration. So during the process of expiration, it's like a balloon, like this. Right. So what happens is that during the process of expiration, since it is reducing the surface tension, whatever tension is exerted during exhalation, that will be subsided by this. So if it is absent, automatically there will be a problem in the process of expiration or respiration, leading to a respiratory distress syndrome. Then comes phosphatidylserine. Phosphatidylserine has main function that it helps in apoptosis. Apoptosis is a process like a programmed cell, that when the function of the cell is over, it is programmed like that. It has to be deleted from the body. It has to be excreted from the body. So phosphatidylserine helps in recognition of that. Then comes phosphatidylethanolamine. It is lining the CNS. So automatically it is again making those functions which are actually liable for the central nervous system. Then comes phosphatidylinositol. It is the most important second messenger. There are basically, if I talk about the second messengers, they come after hormones. Hormones which are the first messengers. Second messengers are cyclic AMP, cyclic GMP and this PI3. So automatically it helps in cell signaling. Then comes cardiolipin. Cardiolipin is the, I would say again, a glycerophospholipid which was first isolated from the heart muscle cardio. Okay, lines the inner mitochondrial membrane, and this is the only phospholipid which is antigenic in action. Now since it is antigenic, so it can recognize the antibodies raised against Treponema pallidum. Treponema pallidum is the microorganism causing syphilis, right? And deficiency of cardiolipin leads to Barth's syndrome. Then we have discussed about the second category. Second category is what? Sphingophospholipid. Abhi what we studied was, we studied glycerophospholipid. So second class is sphingophospholipid. Now see what is the difference here. Sphingo. So basically this sphingo word is coming from alcohol, and the alcohol is sphingosine here. Very, very important. There the alcohol was glycerol. Here the alcohol is sphingosine. Now what is this sphingosine? It is synthesized from the body itself. Body mein from serine. And it is having 18 carbon atoms. It's an unsaturated alcohol, C18, unsaturated alcohol. And if we draw the structure, it is like this: C18. Here OH is there, here NH2 is there, and here OH is there. Okay, so what happens here is this NH2, when it is attached to a fatty acid, this H is attached to a phosphate. Okay, so different components are there. When it is attached to a fatty acid, we call it as a ceramide. Right? And this phosphate, when again it is attached to a nitrogenous base, same thing. Then it is known as a sphingophospholipid. So have you understood the difference? Here the alcohol is different, and this green color is the structure of sphingosine. Like this. which is totally different from glycerol, right? So we call it as sphingophospholipid. Now, if this nitrogenous base is choline, again I will repeat, if this nitrogenous base is choline, we will call it as sphingomyelin. Sphingomyelin. Very, very important. Clear? So, just you have to remember these things, that glycerophospholipid is the one which has alcohol named as glycerol. So, it will be glycerophospholipid. But if the alcohol is sphingosine, it becomes sphingophospholipid, right? And what is sphingosine? Sphingosine is simply derived from serine, obviously in the body, and it is a unsaturated alcohol which has 18 carbon atoms. And whenever this sphingosine combines with a fatty acid, we call it as a ceramide. When it combines with a phosphate, it becomes sphingophospholipid. When it has a nitrogenous base called choline, it is known as sphingomyelin. And as it is clear from the word itself, what is sphingomyelin? Sphingomyelin is the one which is lining the myelin sheath. Lining the myelin sheath. So, it is responsible for the conduction also. It's clear, conduction, excitability, so many functions are there. So, conduction is there, excitability is there. So, this will be the function of this sphingomyelin. Okay, so this is the first class of conjugate lipids, phospholipids, where phosphate is there, where the base is there, and the alcohol can be different. Alcohol can be glycerol, alcohol can be sphingosine. Now the question is, if they are produced in the body, they can have to be degraded also in the body. So if we talk about in general phospholipids, so the enzyme required for the degradation of phospholipids are phospholipases. So I can say that phospholipases is a class of enzyme degrading a phospholipid. A phospholipid. Right. So, the structure of phospholipid is in front of us. Say, I'll just write the structure. CH2O fatty acid 1, CH2O fatty acid 2, CHO phosphate which is connected to a base. This is what we have studied. So, if we talk about phospholipases, so it is going to act here. This will be the first site, this will be the second site, say this will be the third site, and this will be the fourth site. So, if we talk about phospholipases A1, it is going to just release one fatty acid. If we talk about phospholipases A2, it is going to again release the another fatty acid. If we talk about C, it is going to keep diphosphatidyl and phosphate and base will go out. And if D is there, only the base will go out. Right? So, I can say that if attack is on A1, it will release fatty acid 1. If the attack is on A2, it will release fatty acid 2. If the attack is at C, we can say the entire structure will be there. So, I can say phosphatidic acid will be there like this, plus fatty acid 1, fatty acid 2 along with CH2, OCH, and this structure will go like this. And what will be given out? Phosphate, combined with base. And if D is there, simply the base will go out. Okay, so we can call it as, if it is palmitic acid, so we can call it as diphosphatidal. We have diphosphatadic acid, phosphate and base will be given out like that. So this is the mode of action of phospholipases when the alcohol is glycerol. Right, but if it is acting on sphingophospholipids, again it is going to give a different unit. So I can simply write it like this just to make you people understand, because this is very, very important, because in our next video we will be studying about different type of sphingolipidosis. So basically they are the class of lysosomal storage disorders which actually occur in the body due to the deficiency of the particular enzymes degrading it. Okay, so this is phospholipid. If I talk about sphingophospholipid, so sphingophospholipid mein, if we have to just demarcate it, right, I can say that sphingosine plus fatty acid is a ceramide. When it combines with phosphate, it becomes sphingophospholipid, and when it combines with the nitrogenous base, which is choline, it will form pura combined entity will be sphingomyelin. So sphingomyelinase enzyme will degrade them accordingly. Ceramidase enzyme will degrade them accordingly. So this is how we have to learn, right? So this is first class of conjugate lipids, which is phospholipids. Then parallel to this is the second class is glycolipids. Okay, now see, glycolipids also perform the same function, but here what happens is, here lipid is there but no phosphate is there. So the first most important feature is phosphate is absent. Very, very important. Now if phosphate is absent, what is present? So here, if phosphate is present, instead of phosphate, glyco means carb is present. And we have already studied about the classification of carbs. It can be glucose, it can be galactose, it can be oligosaccharides, it can be polysaccharides, or the derivatives of or the sugars. Okay, so mono, oligo or polysaccharides, when they will combine with a lipid, it will be a glycolipid. Now the question here is, what is the alcohol? So always remember, alcohol here is sphingosine. Right. So if we have to simply draw the structure of this, I can simply say that C18OH NH2 fatty acid 1 OH, and it will be attached to a carb. So this structure is ceramide again, but this structure becomes glycolipid. And these glycolipids are further of two types. Right? What are these? Cerebrosides and gangliosides. Now depending upon, main thing is the composition, because we know, as the name suggests, cerebrosides related with the brain, gangliosides, they will be lining the ganglions. Function we have already discussed, right? Now what you have to remember here is that what is the composition? Very, very important. What is the composition of this? So if I talk about, if glucose is there, we will call it as glucocerebroside. If galactose is as a sugar, we will call it as a galactocerebroside. right? Now, why these are important? Because if any enzyme, suppose if I talk about 𝗵𝗵𝗃, the enzyme degrading 𝗵𝗵𝗃 would be 𝗵𝗵𝗃. So, deficiency of 𝗵𝗵𝗃 will lead to accumulation of 𝗵𝗵𝗃. And when these lipids starts accumulating, they will lead to enlargement of a particular organ. And that is what is, it will lead to a disease. So, that order, that you can say those diseases are known as sphingolipidosis. And in general, we call sphingolipidosis as a lysosomal storage disorder which occurs due to the absence of the hydrolysis group of enzymes in the lysosomes. Okay, but this video is just about the chemistry of phospholipids and the glycolipids. Now you must have understood what is the difference also. Phospholipids is further off to, if we have to broadly differentiate between a phospholipid and a glycolipid, I can simply say that phosphate is present, their carbohydrate is present. So in glycolipids, phosphate will be never present, and in phospholipids, carbohydrate will never be present. And depending upon that, glycolipids mein the alcohol is only sphingosine. But in phospholipids, there are two different alcohols. If it is glycerol, we call it as glycerophospholipid. If it is your sphingosine, it is known as sphingophospholipid. Okay, so this is about the phospholipids and the glycolipids, just the chemistry. So in the next video, we are going to discuss another class of conjugate lipids which will include lipoproteins, which is again a very, very important class of conjugate lipids. So this is all in this video. I hope this is clear to all of you, because structures, I would say, even if you don't remember, you should know what are the components. Just suppose if I ask you what is a ceramide, so you should be immediately able to answer ceramide is sphingosine plus a fatty acid. When a nitrogenous base comes with phosphate, it becomes sphingomyelin, because there the nitrogenous base is choline. So the example will be sphingomyelin. Right? So that's all in this video. Thank you so much.